Hello and welcome back, future scientists. I'm Mr. K with Mississippi Public Broadcasting. We're going to continue our lesson today talking about protein synthesis, sometimes also referred to as the central dogma of biology. Today we're really going to look at the first step of protein synthesis, and that first step is called transcription. So let's go ahead and hop into it and learn all the mechanisms that go into this complicated process. So again, what our goal here is that we're making proteins, and so we call this protein synthesis. To synthesize something means to make it. So we're making proteins here. Okay, DNA has all the genetic material that we need. Okay, but the problem here is that it is in the nucleus. Okay, your body is mostly made up of proteins. So because DNA has the instructions on making those proteins, your body's mostly made up of proteins. That's how DNA is controlling how your body basically gets built. Okay, protein synthesis happens in two steps. The first step is called transcription. In transcription, what we're doing is going from DNA to RNA, and specifically it's mRNA, okay? And then once we've made that message or that messenger RNA, we start the process of translation. Translation happens in the cytoplasm, and that's making proteins. So again, transcription is in the nucleus, that's the first step, and then we do translation, that's in the cytoplasm, and that's actually making the proteins. So genetic information flows in this direction. DNA to RNA, RNA to proteins. Okay, going from DNA to RNA is known as transcription, and going from RNA to proteins is known as translation. Okay, so let's talk about transcription in a little bit of detail here. Okay, remember DNA must stay safe inside the nucleus. Okay, so we're going to keep it in the nucleus where it's safe. The ribosomes that make the proteins are in the cytoplasm, so that's where we kind of hit a roadblock, right? So how would you get information to somebody that you can't physically get to, right? Like maybe you've got a friend on the other side of the school. How would you get information to them? You would send them a message, right? Like maybe a text message. So that's what we're going to do here. In the same way, when we're making proteins, the first step is to create messenger RNA, mRNA, okay? This contains the instructions for making proteins and has the added benefit of that it can leave the nucleus, okay? So transcription has three steps, just like DNA replication. You're going to see these three steps a lot. These same three steps are used in transcription, they're used in translation, they're also used in replication. So when it comes to transcribing something, here's what we're doing. The first part is initiation. To initiate something means to start it. If you were to initiate a conversation, that means you start a conversation with somebody, okay? So the first step in transcription is initiation. What happens here is that RNA polymerase binds to a promoter sequence. Okay, RNA polymerase is an enzyme. It's the enzyme that adds those nitrogen bases for us. Okay, it's going to go down the DNA until it finds something known as a promoter sequence. To promote something means to start it. So a promoter sequence here, kind of think of it like a starting line. Okay, when you're running a race, you don't just start anywhere, you start at the starting line. And the same way when we're copying DNA, you don't just start anywhere, you start at a promoter sequence. That's basically the first part of the gene. Okay, the second step is known as elongation. What happens here is that we're using DNA as a template for RNA synthesis. That RNA polymerase moves down the DNA sequence and it adds those complementary base pairs. So we've kind of already talked about these. Remember that in RNA we don't have the base thymine, we use uracil instead. Okay? That's going to continue until we get to the last step known as termination. To terminate something means to end it. What happens here is that transcription, it's reading down, that messenger RNA is reading down the DNA until it gets to something known as a terminator sequence. Okay, that's like the finish line. When the RNA polymerase gets to that terminator sequence, that's saying, hey, we've completed making this gene and we can stop here. Okay, so these are the three steps that are used in transcription. They're the same steps used in replication and translation, just the process is a little bit different. Okay, so what happens next? So in transcription, when transcribing, we're making mRNA. We're making a message, right? Okay, mRNA, like all RNA, is single-stranded. Okay, because of this, we don't make those Okazaki fragments like we did in replication. You might remember when we were making DNA, there was a leading strand and a lagging strand. One strand was easier to copy than the other strand, but RNA is only one strand, so we don't have a leading and a lagging. This process of transcription is easy in the fact that it doesn't have to go back and link all these Okazaki fragments together. So it's made continuously. Basically what that means is it's made in one phase of copying. We start at the start promoter sequence, the whole gene gets copied, and then we get to the terminator sequence, and that's it, okay? When you're making messenger RNA, we use uracil instead of thymine. So anytime you're looking at a sequence, genetic information that's sequenced out, if you're trying to figure out if it's DNA or RNA, if you ever see a T, you know that it's not RNA because RNA doesn't have thymine. We use uracil instead. Okay, so this is kind of a way to represent that information with pictures. Okay, so what's going on here? So look at this little, you see how this kind of looks like a ladder that's on the ground? Okay, well this is DNA. DNA is double-stranded. So what's going on where this black part is, you can kind of see some of the ladders grayed out. But here in the middle where it's black, this is the gene that we want to copy. This is information to make some protein. Okay, so what's happening, this enzyme up here, this is RNA polymerase. What it's going to do is it reads down this DNA until it gets to a promoter sequence. When it gets to a promoter sequence, it's saying, hey, this is a starting point. This is when we can start making this gene. So this RNA polymerase is going to attach to the promoter sequence. Okay, so that would be the initiation part of this whole process. Well, once the RNA polymerase is attached to the DNA, we're going to move into the next step, which is known as elongation. What happens now is you can see part of this DNA, part of the ladder is unzipped, right? And we use that enzyme called helicase to kind of split it open. Well, then RNA polymerase starts reading one side of the DNA, and we're going to add complementary base pairs. That's what this blue line represents. This blue line, this is the messenger RNA, but each little rung of the ladder is a nitrogen base. So we're using our complementary base pairs to find the right nitrogen bases to pair up here. And we're linking all of those nitrogen bases together, okay? This is going to continue until RNA polymerase finds a terminator sequence. A terminator sequence is saying this is the end of the line. This is where you can stop copying this gene. Okay, when that happens, the RNA polymerase disconnects from the DNA. What we're left with, this blue line over here, this is messenger RNA. Okay, you can see that it is single-stranded, and if we were to look at the nitrogen bases, there wouldn't be any thymines. There would be uracils instead. Okay, so this is the whole process of transcription. When this happens, this is going to leave the nucleus, and we're going to start our next step, which is known as translation, which we'll talk about next time. So y'all, thank you so much for making it today. Your teacher has some other work for you to practice on. Make sure to give that your best effort, and I hope everybody has a great day.